Before we begin, I'd like just to acknowledge the traditional owners of the lands where everybody's meeting or at home today, and pay our respects to elders past, present and emerging. For those that don't know too much about the Queensland Academy of Sport, I know we've had people register from all over the world for this event, which is great, and some people will be seeing it via the recording later. The Queensland Academy of Sport is a state-based institute of sport in Australia, and we're focused on four major goals actually. One is to get Queenslanders onto national teams, for those athletes to succeed internationally, mostly in Olympic and Paralympic sports, and then also for us to be recognised as a knowledge hub, so a place where people could come and share knowledge, and also learn from some of our experts in our networks that we have. And then the fourth goal for us is to connect our athletes with the community and to keep that, to try and inspire the community to be more physically active as well. So within the Queensland Academy of Sport, we have this area called Spike, which is where this invitation, you might have got this invitation from, which is Sport Performance Innovation and Knowledge Excellence. And Spike has been going since 2004. Originally it was set up as a Centre of Excellence for Applied Sports Science Research. And then a few years ago, we developed it into Spike, where Spike's goal is to help acquire knowledge through collaborative research projects and student support with university partners and industry. And we also help to try and communicate that knowledge really widely through events like this today. And then also a big focus on translation of research to practice, so how we embed researchers and students within the applied training environments for our athletes. So QAS currently supports about 650 categorised national athletes, senior athletes. And this is our first live webinar for these events. Usually we'd be sitting in our lecture room at the QAS headquarters that seats about 60, and then we YouTube it and share the link. But I'm really excited today that we've got this opportunity, albeit under terrible circumstances of COVID, but I'm glad that we've been forced to use better technology to provide this opportunity to a much larger audience, and then also able to share it later via YouTube as we usually would. So it's with our absolute honour, and very thankful to Ben Desborough and Phil Bellinger and Jonathan Craven today for presenting. And I hope everybody enjoys it. We're gonna, like I said, this is our first event, so please forgive us if things don't go quite to plan. But what we'll do is, if you have any questions along the way, we're gonna hold them off to the end, but we have a chat function in Zoom. So on the bottom of your screen, you should see a button saying chat. If you'd like to pop your questions into there, at the end of the presentations, I'll go through those questions, or the speakers will be able to see those questions, and we'll be able to try and get to them as quickly as possible. So I think that's about it for me. I'm going to hand over to Phil now to start the presentations, and thank you once again for everybody for joining us.
Thanks for that, Jeff, and also thanks to Alan and Brianna for organising the webinar today. So as Jeff mentioned, I'll be one of three presenters along with Jono and Ben, who are also from Griffith University. Myself and Jono are going to present on different aspects of a large-scale training study that we conducted last summer with some well-trained middle distance runners. And we looked at changes in performance, responses to training, and then also the gut microbiome. And then Ben's going to present on some of his related research in this space, and some of the stuff that he's been doing with his team recently, as well as some of the future collaborative studies that we've got planned as well. So the presentations will be around about 12 to 15 minutes in duration, and as Jeff mentioned, we'll just save the questions until the end, or you can type those into the chat function. So there's really been I guess a long-standing belief that the muscle fibre type composition of an athlete is deterministic, certainly in the events that athlete might be able to specialise in. And at least anecdotally up to this point, we also think that this trait might be related to some of the individual responses that we see to training. So I'm going to present some data here that we've collected recently looking at this particular concept. So it's clear that athletes do not have homogeneous responses to training. So that is, in response to a given training block, some athletes may improve their performance, while others may show performance stagnation or even diminished exercise performance. And despite recent research showing that the lack of performance improvement in response to training, which is typically termed non-response, was simply due to not training enough, it's obvious that this paradigm doesn't quite hold true for endurance athletes who already have very high training loads, where the solution to this training non-response is probably a little bit more complex. So this concept is highlighted here in this figure, the bottom left of the slide, by Granada and colleagues, who plotted a series of studies that looked at changes in mitochondrial volume density, which is really a marker of oxidative capacity in the muscle, in response to changes in training volume along the x-axis here. And what this shows is that there seems to be a training volume threshold whereby further increases in training volume do not confer additional increases in oxidative capacity in the muscle. And this might be due to things such as training-induced fatigue and also overreaching. And we've recently shown in our lab where we assessed muscle oxidative capacity using a non-invasive protocol using near-infrared spectroscopy. And we demonstrated that changes in this marker of oxidative capacity were actually dampened in athletes that possessed a high level of fatigue following a very high volume training block, whereas those athletes that were able to tolerate the increase in training load showed substantial performance improvements that were concomitant with changes in oxidative capacity in the muscle. So this table here just describes the different stages of training-induced fatigue, which occur as a consequence of increases in training load, where an athlete may exhibit feelings of acute fatigue, but after an adequate and likely short rest period, this might be followed by either a performance increase or at least performance restoration. If the balance between training stress and recovery is further disrupted, then a state of overreaching may develop, and in some extreme cases, possibly the overtraining syndrome. It's typically considered that inducing functional overreaching may be a normal part of the training process and is often, deliberately induced in situations such as a training camp or an overload training period. Whereas non-functional overreaching is typically considered to be an undesirable state of training-induced fatigue, given the long time course of recovery and often performance stagnation as well. It should also be noted, though, that functional overreaching should also come with some caution. This training state has been associated with impaired sleep quality, which is probably the best form of recovery, a higher incidence of illness in athletes, and it also directly precedes the more severe case of non-functional overreaching, which is undesirable. There's also some recent evidence from others and also our group that have demonstrated suboptimal adaptations in athletes that develop functional overreaching, and that's compared to athletes that did not develop overreaching, but also were able to complete the same relative increase in training load. And it's likely that there's a number of contextual factors that may influence the consequence associated with functional overreaching, and you can see some of those contextual factors here. And I'd probably just like to focus on the last point, which I guess is just appreciating the diversity in the physiological profiles of athletes, even those athletes training for the same sport or the same event. And this was nicely discussed in a recent review by Gareth and Trent, who appropriately suggested that those athletes training for middle distance events, so really high intensity endurance events, can have very diverse physiological and also physical capacities. And it's likely that this diversity can directly contribute to some of these polarized training responses that we may see. So factors such as genetics and muscle fiber type composition and other individual traits are thought to be associated with these polarized responses, but this hasn't really been experimentally shown yet. So the aim of our study was to look at individual responses in response to changes in training volume in a group of well-trained middle distance runners. And we really wanted to identify some of the metabolic consequences that were associated with overreaching, as well as some of the markers of training stress that might precede any changes in performance. So we recruited 24 well-trained middle distance runners, 16 males and 8 females, and you can see their VO2 peak values there. So they were really well trained, and also their season best 1500 metre running times. So if you're not too familiar with 1500 metre event, our male runners here on average are able to post sub-4 minute times, and our female times there were equally impressive. Just to put that into context, they represent around about 16 to 17% slower than the all-time Australian record, so they're certainly pretty well trained. So our study schematic, you can see here, we followed these athletes for a period of 7 weeks, which was broken into 3 distinct training phases. The first 3 weeks was a normal training phase where the athletes completed their habitual or coach prescribed training. And then in the subsequent 3 weeks, we increased the training volume of these athletes by 10, 20 and 30% over successive weeks, where the weekly distribution, type and content of each one of these training sessions mirrored the previous week during the normal training phase, such that it was individualised for each particular athlete. But regardless of which training session they performed during each one of these given weeks, whether that be a Sunday long run or a track session, the prescribed increase in training volume was applied to that particular session. That was followed by the final phase, which was a 1 week taper. So it was an exponential decay model. So overall, that was a 55% reduction in training volume relative to the week 3 high volume training period. And a taper of that nature and duration is supported by experimental and modelling evidence to suggest that it is likely to result in a performance supercompensation. We monitored each training session quite closely. So all the runners wore a GPS watch and also a heart rate strap, and kept a training diary throughout. And the athletes visited our physiology lab before and after each training phase, which I'll detail in a moment, and also visited our MRI clinic, which I'll also detail in a moment as well. So during each one of those laboratory visits, the athletes came in under really tightly standardised conditions. We provided them with food the day before. We also controlled their training for the day leading in to each one of these lab visits, the time of arrival as well. And they arrived in a fasted state soon after waking. We collected a stool sample from them, and also a resting fasted blood sample. We also carried out a number of different physiological measures on the runners. So we assessed their resting metabolic rate as soon as they came into the lab, their body composition. We used our non-invasive protocol to quantify their muscle oxidative capacity. We looked at the physiological response to submaximal running, and also exhaustive running as well. So we performed an incremental running test on the treadmill to exhaustion. So I won't present results from all of these measures, but I will present some results over the following slides in a moment. Lastly, just onto our MRI visit for each one of the athletes. So I've been working pretty closely over the last couple of years with Wim de Rave's team at Ghent University, and also a local radiology clinic, Q-Scan Radiology. But we've optimised a technique here where we can non-invasively estimate the muscle fibre type composition of athletes. And once again, that's a technique that was first derived by Wim de Rave from Ghent University. So we take magnetic resonance images of the lower limb, and we focus on the medial head of the gastrocnemius. And we apply a technique known as proton magnetic resonance spectroscopy, which is a non-invasive technique to look at the chemical composition of the muscle of interest. So we can look at quite a large volume of muscle with this technique. And we can identify a number of different metabolites which resonate at different frequencies along the proton spectrum. And we're mostly interested in carnosine, which we can measure the chemical strength of and obtain an absolute measurement of carnosine. And the reason that we're interested in this particular metabolite is that it's known to be a fast twitch metabolite. That is, it's twofold higher in type 2 or fast twitch muscle fibres compared to slow twitch or type 1 muscle fibres. And we express our absolute carnosine value as a z-score for each one of our athletes in reference to a non-athlete control group, which is age and gender matched. So that being, if an athlete has a higher carnosine value compared to this reference population, they would have a positive z-score. So that represents how many standard deviations that value is away from our mean population. And or if an athlete has a carnosine value that's lower than our reference population, they have a negative z-score. And we think that that negative z-score is a pretty good indication that an athlete would have a higher proportion of type 1 fibres, whereas an athlete with a positive z-score would likely have a higher proportion of fast twitch muscle fibres, or an athlete that sits close to a z-score of 0 would typically have a mixed muscle, so an even proportion of type 1 and type 2 muscle fibres. So on to some of the results. This particular figure here is just looking at the time to exhaustion in our incremental running test, so from before and after our normal training period. So we didn't expect and we didn't see really much of a change there across that normal training phase, and there was a CV in time to exhaustion of just over 5%. And we use that to quantify our smallest worthwhile change in our outcome measure there of time to exhaustion. And we use that to delineate two groups when they came back into the lab after the high volume period. So when the runners came back into the lab on that third visit, so this is after the high volume training phase, the athletes that either had no change in performance or slightly improved their performance but had high levels of subjective fatigue were considered to be acutely fatigued, whereas those athletes that had a decrease in running capacity that was greater than the smallest worthwhile change in the test, they were considered to be functionally overreached. So in terms of the performance responses after the taper period, this is looking at the change in performance from before the high volume period to after the taper period. We can see that there's quite a large individual variation in how the athletes responded and how they're able to adapt to that training phase. And overall, we found that 15 of the 24 runners improved their performance by a magnitude greater than our smallest worthwhile change. And interestingly, the athletes that were considered acutely fatigued had a significantly higher mean performance improvement following that taper compared to the athletes that were functionally overreached, despite some of those athletes also having substantial performance improvements. So we're quite interested in what the contextual factors were that might have influenced the onset or incidence of overreaching, and we didn't find that training volume, either before the study or during the study, was different between our overreached and acutely fatigued subjects. There also didn't seem to be any difference in training status or gender between the two groups, nor was there any difference in age, training experience, or nutritional intake throughout the study, which we monitored pretty regularly just through dietary recall data. What we did find was that there was a significant difference in the gastrocnemius carnosine z-score between groups. So you can see here that the acute fatigue group primarily had quite negative carnosine z-scores, whereas the group that were functionally overreached had more of a mixed muscle profile. So their carnosine z-score was much closer to 0. So what we think that this indicates is that the acutely fatigued group were likely to have a higher proportion of slow twitch or type 1 muscle fibres, whereas our functionally overreached group were likely to have more of a mixed muscle profile, so an even distribution of type 1 and type 2 fibres. Also looking at some of the performance responses, what we're looking at here is the change in performance from pre to post high volume period in relation to the carnosine z-score. So we can see there a moderately strong negative relationship between those two variables. That being the athletes who had much lower or more negative carnosine z-scores and presumably a higher proportion of slow twitch muscle fibres were better able to tolerate that increase in training volume throughout that period, whereas those athletes who had higher carnosine z-scores and more of a mixed muscle profile, they weren't able to tolerate this increase in training load as well. And it's likely that these training responses are multifactorial, but at least some of this variation could be explained by the variation in the carnosine Z-score. Similarly, over here, we're just extending that performance response out to following the taper phase. So it's looking at the change from pre-high volume to post-taper. And we also see a moderately strong negative relationship, that being the athletes with more negative carnosine Z-scores had a greater performance supercompensation compared to those that had more of an even distribution of fibres and a Z-score closer to 0. And then lastly, just looking at some of the training responses and how they differentiated between overreached and non-overreached athletes, we took that resting, fasted blood sample and we measured a whole range of blood biomarkers that reflected inflammation, metabolism, all the way through to muscle damage and iron regulation. And we didn't see any differences in any of these markers between the two groups, being functionally overreached and acutely fatigued. Wellness questionnaires and responses to submaximal exercise were also unable to differentiate between the groups. And we obviously saw changes in the response to exhaustive exercise, which was the metric we used to delineate the two groups. And the overreach group also had a lower peak heart rate and lower peak blood lactate during that test as well. And lastly, we did see some evidence of some differential responses to training in terms of their subjective responses. And I'll show some of that here. So we'll just focus on the two panels over here to the left. This is looking at the training intensity distribution based on running speed. So this is the time that each athlete spends in each one of 3 training zones, which are demarcated by the physiological thresholds. So this is really looking at an external measure of training intensity, so running speed. And you can see that throughout each one of the training phases, there's really no change in the training intensity distribution from a time in zone approach based on running speed. However, when we describe that training intensity distribution based on the subjective responses to training, so that is the RPE of each training session, we can see that there's no real change in the subjective responses in the acutely fatigued group. But the functionally overreached group, in week 3 of that high volume training phase, we can see that they rated a greater number of their training sessions to occur in training zone 3, despite the external load or the running speed of those sessions being the same. So that suggests that obviously the athletes were feeling quite fatigued and they perceived training to be harder, despite there being no change in the training intensity. So in summary, for what we found with this aspect of the study, there was obviously a large variability in the responses to each one of the training phases, in particularly the high volume training phase. And at least some of the variation in that response can be associated with the variation in our estimates of muscle fibre typology. In terms of the markers of training stress, it was really only the physiological response to exhaustive exercise that differentiated between groups, and also some of the subjective responses to training. So in terms of some of the follow-up work that we're doing in this space, we'd probably like to string it back a little bit and have a look at some of the responses to, some of the recovery responses to acute exercise. So looking at how athletes might be able to recover between 2 sessions on the same day. And some of our colleagues in Belgium have done some nice work in this space that was published recently. And we'd probably also like to have a closer look at the taper for these endurance athletes and how we can individualise that a little bit more so. So just before I finish, obviously lots of people contributed to this particular project, and we've got some really good colleagues at Griffith University that have played really important roles in this project, and also others. And also without our strong collaboration with the team at Ghent University and Professor Wim de Reeve's research group, some of these measurements wouldn't be possible. And on that same token, we've got a really good relationship with our local partner here at QScan Radiology, so with both Ben and Hal. And lastly, thanks again to Jeff, Alan and Brianna for organising the webinar today. So next up, I'll let Jono share his screen, and he's going to present a little bit of data on the same project, but more so focusing on some of the changes in the gut microbiome and what all that might mean for endurance athletes.
You see that, Phil?
Yep, looking good, Jono.
Yeah, well, thanks, Phil. Yeah, my name's Jonathan Craven, and I'm currently a PhD student at Griffith University. But I had the privilege of undertaking an honours project with a great research team, and we looked at the influence of training volume on the gut microbiome. And I feel very honoured today to be able to present the results and our findings to you guys today. But before I can do that, for some of you that might not know what the gut microbiome is, some brief background of what it is warranted. So when they talk about the gut microbiome, they might use the term microbiota, which refers to the microorganisms that live in or on the human body. And the microbiome just refers to the cells and genes of these microbes. And they can actually colonize on almost every surface of the human body. And they're actually found within the gastrointestinal tract, with a vast majority inhabiting the colon. So it actually gets you thinking that what sort of impact are these microbes having on host physiology? And actually perform a range of functions. One of the big ones is they actually break down non-digestible polysaccharides, for example, fiber, and they produce these short-chain fatty acids, which they use as a metabolic fuel themselves, but actually can use the short-chain fatty acids to repair the integrity of the gut, which helps in the resistance to invading pathogens. But they can also use these chemicals as a communication system through the nervous system, and they also can synthesize vitamins and amino acids. So the gut microbiome can actually be manipulated and actually takes place right at the birthing process and even during the feeding process during infancy. But diet seems to be the biggest influencer in changing the microbiome. But just recently, stress has emerged. So you've got to think exercise is a form of physiological stress to the body. And it's actually been shown to produce both favorable and non-favorable responses to the gut microbiome. So before I can go in depth any further into our results, I need to go through how we actually characterize the microbiome. So on the left here, we've just got the taxonomy tree for the bacterial kingdom. So they start off at the phylum level and they move down to the class, order, family, genus, then species. And actually, as you work down this tree, it actually increases in an exponential manner. And the literature tends to focus and report changes at the phylum level and then at the genus, but also at the species level as well. They also like to calculate and estimate a person's diversity. So how diversified is a person's gut microbiome? So they use the term alpha diversity, which just refers to the average species diversity within a habitat or a specific area. And they can do this via two ways. They can just look at the sheer number of species and just count how many species are within a particular ecosystem, or they can actually look at the number and abundance. So looking at the spread and evenness of the species, which tends to be a better indicator of a person's gut microbiome diversity. They can also use the term beta diversity, which just looks at different species composition between sites. So looking at the respiratory compared to the gastrointestinal site. So what do we know about athletes and gut health? Well, the pioneering work done by an Irish research group first looked at elite rugby union players, and they compared it to a control group that was matched for age, sex, height, and weight. And what they found is these rugby players had a higher gut microbial diversity when compared to the control group. But what they also reported was that they found a positive correlation between protein intake and gut microbial diversity, and which the rugby players were consuming far more amount of protein when compared to the control group. And another study actually found a positive correlation between aerobic fitness and gut microbial diversity. But these two studies were cross-sectional in nature, so they didn't track the gut microbiome over time. They just looked at it at one time point. But in saying that, a recent study has emerged that looked at elite race walkers after performing a period of intensified training for 3 weeks. So they looked at a between group difference. So one group consumed a low carbohydrate, high fat ketogenic diet, and the other group consumed a high carbohydrate diet. And what they found is both groups had no change to their alpha diversity, but what they did find is some subtle changes in a few genera, but this was only observed in the ketogenic group, not the high carbohydrate group. So it's not necessarily the intensified training that induced these changes, more so driven by diet. And furthermore, some recent studies that actually use quite extreme exercise interventions, they actually found an increase in alpha diversity. But these two exercise interventions were, one was a 5,000 kilometer transoceanic row for 33 days, and the other one was a 4-day military ski march during the Arctic winter. And that military study actually found an increase in bad bacteria, while they reported a decrease in good bacteria. So the increase in alpha diversity was not a positive outcome in this particular study. And I just want to highlight that these two studies are really not reflective of normal training. So what don't we know? Well, what's the impact of training volume? There's only been one study, and it was cross-sectional in nature, and they looked at professional and elite amateur cyclists. And what they observed was that the more hours that was spent training, those particular athletes had a higher abundance of Prevotella, which is a beneficial bacteria. But Prevotella is also being linked to a long-term carbohydrate diet, which is consistent with these type of endurance athletes. So we really do need to get a deeper understanding of what the impact of training volume is on the gut microbiome. And that's why we sought out to examine a realistic response to an increase as well as a decrease in exercise training volume. Now, Phil went over the study design in quite detail, so I'm not going to go over it again. But the gut microbiome analysis was done in a subset of participants. And the reason being is we wanted to exclude any participants that had a poor record of their dietary intake, because we measured diet pre and post each training volume. So that left us with 14 runners. So they were of similar age, VO2 peak and training status to the subjects that Phil went through earlier. So as I said, we looked at diet. So we analysed the runners' diets, and 2 investigators completed the analysis. So we looked at both macronutrient intake as well as energy intake and fibre intake. And we actually found no significant change across time for both absolute and relative macronutrient intake. And that was the same for energy intake as well as fibre intake. And we were interested in fibres because there's been a positive relationship reported between fibre intake and gut microbial diversity. So we actually found no changes in any dietary parameter. And over the right here, this is just the between investigator analysis of the diet. And I'd just like to highlight that these runners we observed them in a free living. So they were free to consume the food as they normally would. We didn't control the food outside the standardized dinner and breakfast before the exercise trials. So now moving into the microbiome results, we're going to start off looking at the alpha diversity. So on the left here, we're just going to look at the species richness. That's just looking at how the number of species within the runner's gut microbiome. So we actually found no significant difference post high volume training when we compared it to normal training, as well as comparing that to taper training for that matter. And then when we looked at both the species richness and abundance, again, we found no increase or decrease post high volume training. And the same was for taper training as well. We then went to look at some relationships. So looking at the relationship between alpha diversity and VO2 peak, as well as protein intake. And we found no significant relationship with either parameter. Then moving on to the taxonomy, we wanted to start looking at the changes at the phylum level. So remember, that's at the top of our bacterial kingdom. And so on the y-axis here, we have the relative abundance in percentage. And then along the x-axis is just the individual runner's data for the corresponding training time point. So we found no significant change in any phylum across time. But what we did observe is that the Firmicutes and Bacteroides were the most abundant phylum within this cohort of runners. And that's consistent with the literature for both an exercise population as well as a sedentary population. But also, despite recent research characterizing athletes' gut microbiome, this graph just highlights just how individualized the gut microbiome is, especially for an endurance cohort such as this that has similar training characteristics. We then wanted to have a look at if there was any changes down at the genus level. So we first wanted to look at the top 15 abundant genera within this cohort of runners. And again, we found no significant change in any of these top 15 abundant genera. So this colorful graph just highlights again just how individualized and diversified the gut microbiome is within an individual and comparing it to another individual. But in saying all that, we did find some significant changes in less abundant taxonomy. And I'd just like to start down the bottom here with Haemophilus parainfluenzae. And the reason being is that belongs to the genus Haemophilus. Heme meaning iron. These particular bacteria love iron and need that to grow. And this particular genus belongs to the family Pasteurellacea, which belongs to the phylum Proteobacteria. Now, Proteobacteria are known to have pathogenic bacteria belonging to that particular phylum. And actually, Haemophilus parainfluenzae is a known pathogen within the body, which has been linked to endocarditis, bronchitis, as well as pneumonia. So we actually observed a significant decrease following the high volume training period. Now, this is the first time I'm aware of seeing such response with this particular bacteria. So more research is warranted to see if other exercise interventions have a similar response when it comes to this particular species. We also found a significant decrease post high volume training in the genus Lachnoclostridium. Now, this particular genus is known to produce acetate. So acetate is a short-chain fatty acid, and high levels of acetate have actually been shown to stimulate the parasympathetic nervous system, which can go on to stimulate glucose, insulin secretion, and granulin secretion, which actually can modify eating behavior. So further research looking at the decrease in Lachnoclostridium in context of both diet as well as different training interventions is warranted. And then lastly, we found an increase in the species, Ruminococcus colidius. Now, this particular species belongs to the genus Ruminococcus, and they're known to break down polysaccharides, but this particular species cannot, and it likes to digest and metabolize simple sugars such as glucose, fructose, and galactose. So, and we actually found a significant increase in this particular species, but we also noticed an 18% increase in simple sugar intake during this period. So one could speculate that this increase we observed was potentially due to the increase in simple sugar consumption. But what was very interesting and what we observed is post the 1 week taper period, none of these changes that we observed post high volume training returned back to the normal training levels. So what does this all mean? Well, an increase in training volume appears to not influence the alpha diversity. However, it can produce some shifts in less abundant microbiota, and a 1 week taper appears to not be enough time to return these minor microbial shifts back to pre-high volume training levels. So how's this applied to athletes? Well, they can take comfort in knowing if they engage in a period of high volume training, a realistic engagement in high volume training, that's not going to be accompanied with a detrimental impact to their gut health. So where to from here? Well, we looked at training volume. What about alterations in exercise intensity? What about different lengths of taper and different sports? Now, we looked at a high impact sport such as running. What about a low impact sport such as cycling and swimming? And then finally, looking at different climates. So what's going to happen if an athlete exercises in a warm climate, if they have to prepare to compete in a warmer climate? Is there going to be a detriment to their gut microbiome and subsequently their exercise performance? So that'd be interesting questions to answer in the future. So I'd just like to quickly acknowledge the Queensland Academy of Sport for the funding, as well as Griffith University. But our research team, especially Dr. Phil Ballinger, as he spearheaded this entire project and worked tirelessly hard for the 4-month period. But finally, the participants, they were a great cohort of runners and they were a pleasure to work with. So thanks for your time, and just like to pass on to Associate Professor Ben Desborough now.
Okay, cheers, Jono. Okay, how does that look? Okay. All right, so first of all, we're going to change tact a little bit here from the point of view that Phil and Jono have given us a very detailed look at a very large study that was run within the group down here at Griffith. And it's been a great pleasure to be involved in that project. The next sort of 10 minutes, 15 minutes or so, I'm just going to give a brief overview of a range of studies that we've been conducting down here in the area of acute recovery. And so the focus of this work has been really to look at nutrition strategies. Being a dietitian, that's my area of interest, as to how to improve a athlete's ability to recover when they've got a limited period of time between exercise bouts. And really the focus has been, probably in less than 8 hours. So you've got a restricted time window with a focus on the subsequent exercise bout being a performance-orientated bout, as opposed to doing some training where you might be focusing on skill or adaptation of metabolic changes, but really looking at, well, how do we maximize nutritional recovery on the basis of then moving through to a second exercise bout. And obviously that can occur within a training to training bout, and they could be the same type of training. They could be different modes of training within the same athlete. Equally, it could be from a training phase to a formal competitive phase. We have acute recovery, obviously, between competitions as well in different sports. So that 8-hour period of time can also involve a range of other factors. So there can be travel. We can have episodes of sleep. We've got lots of athletes who have a job and they will have employment commitments. And then we also need to consider the social context that the individual lives and operates within that sort of period of time, because that may influence their dietary patterns. I think we also need to consider that after one exercise bout, it's not as simple as then switching straight over to eating and drinking. This is a photo that I recently got from a friend and colleague, Greg Cox, after a triathlon that he attended in Japan. And you can see here that this young lady who's in a recovery pool looks incredibly stressed and is highly unlikely to be consuming large volumes of food, at least in the next sort of 5 to 10 minutes of time. So we can put in as many strategies as we like and have a great understanding of nutrition. But if the athlete is feeling like this young lady, getting them to consume large amounts of food at that particular time is likely to be compromised. So if we look at recovery from a nutrition perspective, I really like this diagram from Jan Lemur, which highlights the main areas that we're interested in. So refueling, restoring carbohydrate, repairing, looking at muscle damage and repair. So that's largely a protein-mediated effect. Rehydration, looking at replacing lost fluid, and then obviously relaxing and having the athlete not necessarily expending excessive amounts of energy, mentally relaxing as well between those acute exercise bouts. Now, our research area, or my research area, really initially focused around looking at replacing fluid quite quickly. So what I want to do is talk to you about a couple of the studies that we've done in this area over time, and then highlight where we're going to in terms of our sort of future work. So when we start to look at rehydration, you go back into the literature, the studies that typically have given us our information around rehydration have been very structured studies where we've controlled lots of variables and then looked at one particular factor within different beverages. And we've done some of this work, but we are only a part player. There's been lots of other research groups around the world who've looked at this. This is an example of one of the studies that we looked at, where we looked at milk and different types of milk on acute rehydration after an exercise bout that's caused a degree of dehydration. In this particular study, we looked at cow's milk and we compared it to soy milk. We also looked at comparing it to Sustagen Sport, which is a milk-based liquid meal replacement, and then we compared it to Powerade, a water-based, obviously sports drink product. So we've looked at different nutritional profiles of drinks. We've also gone into the dark side and had a look at beverages that are popular in that post-exercise period to see things like the influence of electrolytes, the influence of alcohol, and some of these studies have taken us to sort of very dark places. If you collectively look at this data, this is a table that we put together for a textbook last year, which has combined a lot of different studies that have looked at different beverage types. And you can see here I've highlighted the Beverage Hydration Index, which is a collection of work on a variety of fluids, which has attempted to standardize the influence of consuming different types of beverages post-exercise. And broadly speaking, we group them in three categories. So if we compare things to water and drinking water after, well, a beverage hydration index isn't necessarily after exercise, but it's standardized to water. If you drink a sparkling water, if you drink a sports drink, if you drink a soft drink type beverage, tea, coffee, and low alcohol beer, you'll probably retain, within the context of a structured environment where you just give a fluid after exercise, a similar amount of fluid from those beverages that you would if you just drank plain water. If you want to retain more fluid within the context of this post-exercise only giving fluid scenario, then most of your milk-based drinks are going to see more of that fluid retained. And then if you use oral rehydration solutions, you're equally going to see some additional fluid retention from those products. And if you drink beer and the beer is full strength or greater than probably 4% alcohol, you're going to see some of that fluid lost. So this is within the context, as I said, of sort of very structured, rigorous protocols where we are giving a fluid exclusively. One of the challenges with these studies has been that we give fixed volumes of fluid. So when we're looking at a different type of beverage, we might say we'll give you 100% of the fluid that you've lost after your exercise task, or perhaps 150% of the fluid you've lost after your exercise task. But individuals drink different volumes of fluid, and some of that's to do with how full a beverage makes them feel. Sometimes it's to do with how big their water bottle is and how much fluid they have available to them, or what commercial sizes beverages are sold in. We wanted to have a look at this. This is a study that we looked at comparing how much, in a voluntary sense, so ad libitum, somebody consumes when you give them a milk-based supplement, so Sustagen Sport versus Powerade, to have a look to see when people are given a free choice of volume to consume, how much do they actually consume. And we repeated this trial. So what we have here is two Ps, P1, P2, and two S's. So this is Powerade and this is Sustagen. And I really want you to concentrate on this area in here. So this is the fluid loss during exercise, and then they're given exposure to one of these beverages. So they don't get both, they just get one on the different trials, and we wanted to have a look at repeatability. And you can see here that the circles are the two Powerades and the triangles are the two Sustagens. And so the first thing you'll notice is they're pretty close with one another. They actually replicate pretty well from one trial to the next. But it's only if they're given access to the water-based drink as opposed to the milk-based drink do they actually drink enough fluid that potentially they may replace all of the fluid that they've lost. So whilst the milk beverage is well retained, typically in an ad libitum sense, people in this study didn't drink enough to replace the fluid that was lost. Now they went on to squirt out a lot of that fluid, mind you, which is a totally separate issue. But if you just look at replacing fluid loss, then that study suggests that a water-based beverage is going to be consumed voluntarily to a larger extent than the milk-based beverage. We've also recently published a study looking at how quickly people drink and whether that has an influence on how much fluid is retained. So the first part of this study we conducted in Australia, and we collaborated with some international partners in Scotland to have a look at a sort of second phase. So if we just have a quick look at the data from that study, in the first part that was conducted in Australia, we compared milk, and it was just regular cow's milk, to water. And the 30 and the 90 relate to how quickly the person was told to consume the volume of beverage that they were given. So on one trial, we have water that was consumed quickly over 30 minutes. On another trial, we had milk that was consumed quickly over 30 minutes. And then the two slower trials with the two drinks. Now, these graphs are cumulative urine output. So really the key thing is what's happening at the back end here, because this is fairly close to when people have consumed the fluid, and this is obviously the accumulation over time. Oh, pardon me. I'll try and go backwards. And what we can see here is that the two, I've gone one too far, sorry. My computer's just running a little bit slow. Here we go. So what we can see here is that these two circle trials up here, where you're seeing large amounts of fluid being lost or cumulative urine output, are both the water trials. And there's not a huge difference between whether they drank the water quickly or drank the water slowly. And whereas these trials down here are our milk trials. So the water, sorry, so the fluid retention from the milk is far greater, irrespective of whether they've consumed the milk quickly or slowly. Now, because the separation we had here was quite clear based on the type of beverage rather than the rate, our collaborators in Scotland, we decided to speed up the drinking rate a little bit. Because if you ask someone to consume a drink over 10 minutes in our lab, we see some people drink all of that volume within 30 seconds, and then they wait the next 9.5 minutes for the next aliquot of fluid. Whereas other people will measure their drink out over a long period of time. So people can drink large volumes of fluid very, very quickly. Other people tend to space their fluid consumption out. So 30 minutes to me is still relatively slow drinking in comparison to some of the things that we see in our lab. So we looked at 15 minutes, 45 minutes, and then a repeat of the 90 minutes drinking. So these are all water now. So we didn't consume milk in the second arm. We just wanted to have a look at the rate of drinking. And you can see here that there's actually no difference between any of these trials, between the 15, the 45, and the 90. There's a slight separation there from the 90, but that's largely because they haven't consumed all of the fluid until 90 minutes, which is about here. So it's sort of catching up. And this variance that we had, with some repeatability work done, that variance is no greater than the day-to-day variance that we were able to see in some of the repeat trials. So there is actually no difference between whether you drink water within 15 minutes, and this is 100% of the fluid that they've lost, as opposed to sipping it slowly over 90 minutes. So you don't need to necessarily be too concerned about drinking rate. I like to put things in terms of Twitter to summarize. And if we have a look at the summary from that work, is that fluids in milk, or milks, different types of milks, are well retained post-exercise, but voluntarily consumed in small amounts. And the drinking fast, and in this instance, fast drinking is about 15 minutes, or slow, doesn't impact on that fluid retention. So the body is primed after dehydration to try and retain fluid as quickly as possible. And unless you drink huge amounts of alcohol or high concentration of alcohol, it's going to do a pretty good job at trying to retain that fluid, even if you drink quickly. Our more recent work, and probably the more interesting applied work, has really looked at a combination of both rehydration and refueling. Because it's very rare that within an 8-hour period, or within a 4-hour period post-exercise, people only consume a drink. More often than not, they'll have something to drink, and they'll either then have something to eat, or potentially be sleeping and not consuming fluid over a long period of time. So we've now done a number of studies that have looked at ad libitum consumption of fluid and provided food which people can consume ad libitum as well. And the first study that we completed was completed in 2017. It was in males, and then we had a look at a female population because we wanted to compare the results. Men eat differently, have different beliefs around food, and we wanted to explore that. And then the final study was a combination of both males and females. I'm going to show you the data from just one of these. It's actually the first study. And the reason I'm showing you that is that the results have been pretty consistent across the 3 trials. And so this is what it looks like. What we have here again is dehydration caused by exercise, and then consumption ad libitum, and that's why they don't come up to all of the same spot here, of different beverages. So in this first study, which was the male study, we actually did a repeat water trial. So we only gave them access to water post-exercise, but on 2 occasions, they also got access to a suite of foods to consume, and they could consume as much or as little food as they wanted. On a 3rd trial, we had a Powerade, and on a 4th trial, we had Sustagen. So they were only given access to one of these beverages, but on 2 occasions, from a repeatability perspective, we repeated the water trial. So what you can see here from a net fluid balance is that at the end of the 4 hours, when they've had food and fluid, all of the drinks pretty much end up replacing the same amount of fluid. The person may drink more, but the body does a very, very good job at retaining that fluid to the point where it's trying to recover fluid balance. And if you look at USG, or if you look at plasma osmolality, across all of the trials, they're fairly consistent. So the body, when fluid is combined with food in an ad libitum context, does a great job in trying to restore that fluid quite quickly. What we have been able to show, though, is what is the nutrient intake when you combine the fluid and the food data to look at the total nutrient package. And this is quite interesting from the point of view that we have a total consumption here. So the total size of the bar here is the food and fluid combined. But obviously, when you're giving water, you're not bringing in any calories from the drink, whereas you're bringing in drink calories when you're drinking a Powerade or you're drinking Sustagen Sport. And obviously, if you're drinking Sustagen Sport, you're bringing in a lot of calories. You're bringing in fewer calories, but still substantially more than the water trials here. And what you can basically deduce from this is that the calories that are provided in the drink, whilst the person eats less, are never fully compensated for by a reduction in food intake. So in other words, the calories from the drink combined with the calories from the food typically result in a total intake of calories which are far greater than if you had no calories in the beverage at all. And we've seen this across all of our trials, that the calorie density of the beverage has is reflected in the total calorie consumption. So whilst they might eat more following consumption of water, they never eat the amount which would offset the calories that are in the drink. So in terms of summary, from a rehydration perspective, with food, from our data, all beverages become equally effective. But from a refueling and repairing perspective, the drink and protein calories count. So if you're interested in increasing carbohydrate intake, increasing protein intake, then the use of a calorie or protein containing beverage is critically important, because you're going to provide more in that context. The other area of research that I'm particularly interested in is the translation of sports nutrition advice to the general public. And we have to remember that most individuals who are receiving our advice aren't doing exercise sessions that are separated by 4 hours or a maximum of 8 hours, and then are ongoing training day after day. They're often training once a day, sometimes 3-4 times a week is what they're interested in training. So from that perspective, the advice to consume a calorie-containing beverage may be completely unassociated with what they're trying to achieve by their exercise task. And so in those instances where the person's got access to food and they're just trying to maintain a healthy body weight, drinking a calorie-containing beverage in that context is actually undesirable. So I think we need to be very careful as to the context in which we provide our advice. So our next phases of this research, you've met Jonathan already. We have another student, Jennifer, who's online at the moment, who's just joined us from the UK. We're very interested in exploring these areas in more detail. This is an example of the sort of study design that we're looking at for the next phase for some of Jonathan's work, where we're interested in having a look at the influence of nutrition on sleep, but in particular disrupted sleep. So we're looking at study designs around, can we come up with a nutrition protocol that has a profile of nutrients that might be perceived as being of benefit to improving sleep quality, and in the context of somebody who has disrupted sleep or restricted sleep, can we improve cognitive function? Can we improve physical performance the next morning via a nutrition mediated modality? And so looking at combinations of sleep disruption and nutrition strategies. And Jennifer joins us, as I said, from the UK, and she's got a particular interest in relative energy deficiency in sport. And the first phase of work that we're looking at here is to look at how coaches are supported to develop our understanding and management strategies around relative energy deficiency in sport and low energy availability, but particularly in younger adolescent athletes. So we're going to be developing some protocols in that space as well. Thanks very much. Okay, so I might go back to Jeff.
Okay, thanks, Ben, and thanks everybody for the questions that have been coming through in the chat function. I see that Phil and Jonathan have been madly typing and answering a lot of people's questions, which has been great. And I really encourage everybody to have a look at the chat function in the Zoom chat, because you'll be able to save that chat as well. There's a little button down the bottom right that says file, and a couple of dots next to that. You can save that chat if you'd like to review some of the questions and the answers that have been provided. If there's any other questions now, though, there is the ability to raise your hand in this forum. I'm sort of testing our ability here with the technology. Otherwise, please continue to pop them into the chat, and the presenters will try and answer them within the chat. Otherwise, that's the sort of end of the formal presentations that we had, but we have allowed some time now for those that are still online, if they'd like to have some discussion with the researchers. The only thing we've got to be careful of is that not all 100 people with their microphones turned off suddenly get turned on. And so we might try and do that through the raising the hand function or through the chat function. So if you're happy with that, Ben, I think we might just keep proceeding that way.
Yep, that's fine. I've just, I see a question there about sharing a Twitter take home. I'll just put that on the text. I won't go back to the slide because then I'll be mucking around with slide sharing, but I'll just put the text that I had for that tweet just in the chat box. Okay.
Yep. And Ben is recording this, this webinar as well. And so it's our intention, with the presenter's permissions, of course, is that we're going to try and upload this presentation up into the QS YouTube channel. And so please keep your eye out for that, and we'll send out a link to everybody who registered for the event so that they've got a copy of it if you'd like to go back and review it. But apart from that, on behalf of the QAS, we're really, really thankful for the great presentations, and also for everybody for taking the time to log in during this challenging time, and the great questions that are coming through the chat. So, so please stay online and ask and answer some questions on the chat. Keep the chat going. Otherwise, please keep your eyes out too for the next Spike events that are going to be coming up on the webinar.